So hello and welcome. My name is Bernie Lee. I'm the product manager for LabKey Biologics LIMS. It's a product used commonly for the development of therapeutic antibodies or other large molecules, ligands, cultivated meat, just to name a few. And today I'm here with a short presentation to share some of the investments that we have made and are continuing to make in our product, LabKey Biologics LIMS, to support discovery operations. So we make a point at LabKey to build long-term relationships with our clients and industry collaborators that really allow us to learn firsthand what sorts of data management challenges are present. Many organizations developing biologics, such as therapeutic antibodies, do a great deal of their own head of pipeline work and have expressed to us their challenges for discovery. Many of the most common pain points we've heard relate to the extensive use of spreadsheet work across lots of files, shared or individual. These can be very inefficient operations that carry a high risk of human error, which I'm sure you're all familiar with. And they provide no integration across data types. So that's no connection between results, samples, plates, the biological entities you're actually analyzing and engineering. It all has to kind of be done by hand. And of course, there's poor traceability, availability, and controlled access for the data in these files. There are also pain points arising from a lack of structured, centralized data management. So this slows down coordination and decision-making, as information has to be gathered by multiple people and recapitulated for use. It's also extremely taxing to clean it for informatic use, such as feeding artificial intelligence and machine learning algorithms, and for other programmatic means. And it's similarly painful when you finally arrive at a regulatory submission or some other aggregate data use scenario to pull all that data together for that purpose. And the third category that we hear a lot about are pain points arising from organizational challenges. So this is scientists, managers, and executives all having difficulty tracking the use and storage of your samples, materials, plates, and other such things between workflows, between projects, between teams, between locations. And there's also a loss of knowledge that can come through staff turnover, since often, and especially with spreadsheets, only they are intimately familiar with their data and exactly how it's organized. Now, these are problems that we solve all the time for other scientific endeavors, but had not yet done so for common discovery operations. That's why today I'm very pleased to announce support for these workflows. Biologics LIMS will now bring greater efficiency and faster decision-making to antibody discovery by centralizing data operations, integrating samples, plates, and assays, along with various biological entities, and automating routine analyses, all in a structured, high-utility way. So this includes support for plate set configuration, for sample management throughout your campaigns. That's for plates being moved through screening rounds, stock plates that might end up in storage, plates being prepared for assays, automating data capture and integration with samples from plate-based assays, automatic routine analysis generation. So that includes reports and visualizations that might be needed to help characterize and screen, and then select your best candidates. This includes support for centralized integration of all data through multiple rounds of screening and characterization, enabling easy tracking back to your clones, sequences, molecules, and other source entities. And of course, programmatic access to all the structured data, facilitating that informatics usage, because of course, we're all trying to improve our library design and predict developability risks, among other things. So there are the integrated feature areas that form the cohesive cloud-based application that powers the capabilities I've just described. and help alleviate some of the challenges faced by early stage biotechs. Those areas include sample management, which again, in general, helps track the lineage and usage of samples and plates. Our bioregistry, which is where you can define and track all of your various entities and sources related to your campaigns in a central repo. Our integrated ELN, so you can document experiments, highlight results, assay data management in general, whether it's plate-based or not, to capture results for rounds of work and automatically integrate them with samples, and our workflow manager, which allows us to create task-based workflow templates to match lab processes and guide data collection, as well as facilitate handoffs between staff. Now, each of these feature areas are capable of a great deal more than I can show you today. We have, of course, previous webinars and other materials to help people learn about these. However, each of these are adaptable to the needs of your organization, and I certainly welcome the opportunity to demonstrate them to you at a future date with the requirements of your organization in mind. Today, however, I want to keep the discussion short and focus on showing you a few screens representing some of the kinds of things newly supported for discovery. So you're going to see information captured and relationships between the sequences, molecules, and libraries that you may have in your inventory of glycerol stocks, plating and metadata capture for intended screening and characterization work, analysis and selection from your candidates and leads, and authorship of data-connected notebooks narrating the operations. Please note that these are configured for generalized use, and again, can be tailored to your unique needs. So here, let me switch over here. Sorry, one second. There we go. Apologies. Okay. Hopefully that's coming up. There we go. So, to start with, I just want to show you this mature view of how different types of data in the system are related. You can, of course, see how nucleotide sequences are combined with whatever vectors you use, and go into some kind of expression system, whatever the type is that you in particular use, and they produce some molecule which is composed of a certain set of protein sequences, and downstream those are in the form of samples which you can use in and out of plates. And when you first start a campaign, you may only have information about your libraries and stocks, but as you learn about your sequences and then produce and use them in a variety of ways, all of this information is ultimately integrated together for you. Now, here we go. Here, I can capture information about my libraries. In this case, it's just some basic information about their type, their diversity. These libraries each have stocks that may be available for use, and I can go over and show you some of those. So, analogous names to show those relationships by convention here. And in this case, we can see that these particular samples, these particular stocks, are plated by yours truly, and it's easy for me to choose those samples, and I can either plate them or pool them. So depending on how you're operating. So in my case, I can pool these down and then plate them, or I can select all of these things and I can create a new plate with their contents. Now, in this case, I already have a plate pre-configured with that information. Here are those same samples on a pre-configured plate where I have captured where I have captured metadata about these. And yeah, so in this case, really basic information: concentration, diluent volume, and what well type these actually are. But of course, I can capture whatever additional information is relevant in this case. So I can pretty easily grab and capture information about antigens or basic description. And once these are plated, it's easy to capture data against it. So I have a couple of assays in here pre-configured. I can just click in here and take my plate read and throw it against this plate. Again, I'm not going to walk through the plate reader import necessarily, but I do want to mention that in addition to being able to just manually associate these things, you can automate these through our file watchers that listen for reads that come off of instruments, or can be dropped in a designated location, as well as through APIs if you happen to have a pipeline set up. So in this case, I'm going to again just jump into a pre-imported ELISA, where, as you can see, the same information about the plate, well locations, the samples are available, and now we have the optical density measures that come from the plate read. We automatically calculate the ratio, in this case, of that optical density over baseline, which you can then use to select samples for additional rounds of work. We also automatically generate a few visualizations, which can be customized. So in this case, I can get a histogram view of these different measures as they come in. I can also get a heat map of my plate, so the optical density values that come in per location, and I've prefabricated some views of, in this case, just the best samples in here, in this case 4x over baseline, which we then automatically filter the grid for. So if you're only interested in that 4x over baseline one, that's what's going to come through. Now, once we have these filtered, and I just want the top actors, I can easily select those and then say, put these in a new plate, allowing me then to propagate forward just the materials that are of greatest relevance and continue to characterize and select them. So in this case, again, I already did that, and I created a plate that I'm going to use for some more advanced functional characterization, and I went ahead and imported some alpha ELISA data against that plate. In this case, I actually just did this against 3 of the best performing samples, and I plated them at 10 different concentrations in triplicate, which allows me to do things like calculate the EC50 and max values for these samples, and automatically generate curve fits for these. In this case, I've done a 4PL curve fit, and we can see the values that are most relevant. These things are all configurable. So if there was a different visualization relevant or a different calculation that was needed, those things can all be done on import of this data. And in that way, again, we can really automate those routine analyses. Now, as I said before, as you progress through these rounds of work and generate more data in our system, it all gets connected together, ultimately kind of building that tree that I was showing before. So if we look at this particular sample that came from this particular library that ultimately produced one particular molecule, I can actually come in here and say, okay, I've gone through several rounds of work. I know ultimately I've been looking at at this particular molecule or this particular sequence. Show me all the data that I have for this, and we aggregate it all for you. So this data should look familiar because we were just looking at it. This is that alpha ELISA data that we had for this particular sample. We have the original ELISA data that we used to do some initial screening and select it for the additional functional characterization, and the original titer data for this information that was used to plate that ELISA. Okay, so that's higher order questions. The other thing that I wanted to briefly mention is how this all can be woven together as part of a data connected ELN. So here I have a very simple notebook write-up that captures some basic information about candidate selection profile, and I've put in some very basic protocol steps for doing some initial phage panning, some initial enrichment. And as you can see, I have the steps I've taken on an initial plate, which I could click into and view the samples and metadata for. And through each successive round of screen and enrichment, I have the new plates if I replated them, and the steps that I took as I proceeded. And of course, at the end of those rounds, I can capture the quantification that I did and showed you. I can capture the selected samples that meet my criteria, and describe the next set of steps that I'm going to do here. And of course, you'd have a lot more write-up about your methods and stuff. But in this case, this is just a simple demonstration of how you can weave together the structured information that you have captured, as well as the unstructured narrative of what you did, improving reproducibility and allowing you to simply share these things with people making decisions about your projects, or ultimately aggregating this information for regulatory submissions and other use. So I'm going to go back to my presentation here. Perfect. And kind of sum this up a little bit. So, I want to try to simply state that with our new support for these discovery workflows, we want to help you reduce your dependency on spreadsheets for data management, increase the efficiency and throughput of your discovery operations by tracking related samples, plates, and entities, and the analyses as they progress through these multiple rounds and campaigns. Establish and maintain those data relationships, entity uniqueness, auditing, and controlled access. We want to help you centralize that data capture, organization, and management for faster operations, and meaningfully group data by project, by workflow, by development stage and team, and document experiments and processes by aggregating structured and unstructured information. Again, I said I was going to try to keep this relatively short, and so I really appreciate your interest in our work, some of your time today. We would really like to learn how well this new offering serves you and supports your unique research. So please get in contact with us for a deeper demo, conversation, and trial. And we definitely have a few minutes. And so if anybody has questions that I can address in this forum, I would be delighted to do so. Thank you again. Oh, and in the chat, we have added a link that you can follow to get in touch with us, schedule some time, help us to learn from you and see how good of a fit we can be for you. Okay, we do have one question. Okay, so the question is about how our Biologics LIMS is made available. And it's actually available two ways. We offer it cloud hosted, which means you don't have to do any of the work. It's simply made available to you. We also offer it on-prem. We know that a lot of biopharma, biotech organizations need to have their own infrastructure and have very tight control over these applications, these servers, and so we do allow for that. We do provide for that. And there's a question in here as well about where the data is stored. And in this case, it depends on which of those avenues you choose. So, all of the data is structured in a relational database, and like I said before, is accessible via API, as well as through our application. If you host it yourself, that would be on a database that you hold and maintain. And if we offer it to you in the cloud, that's data that is exclusively yours in your own dedicated and secure cloud environment. Let's see. One more question came in. Thank you. Let's see. How does this integrate with other types of tools? I mean, I guess the question is a little bit more specific than that, but if you don't mind, I'm going to answer a little bit generally. And if we want to dive deeper into some specific tools, I'd love to have that conversation individually. So yes, our Biologics LIMS integrates with a variety of other tools, whether those be instruments or whether those be analysis tools or data pipelines. You can connect other systems to our Biologics LIMS either through direct database connections, through APIs, and we do have a variety of direct integrations with, say, sequence analysis tools, things like that. If there's one in particular that you're interested in talking through, please let us know, and I'd love to have that conversation with you. Let's see. So we got one more in here that's about some particular assays. Yeah, so the question about which assays are supported is essentially the question. So what I showed you, I had essentially 4 assays configured, as you saw. I think I had a titer assay, I had ELISA, alpha ELISA, and one that was for inhibition. We support almost any type of assay data capture. I simply configured for. Some of our existing biologics users have upwards of 40 or 50 different types of assays for different purposes that come in different forms and formats. We can support just about anything, and ultimately it results in the same kind of interaction that I showed, where you just say, I want to import against this assay type, and you can throw your file at it, and we will automatically parse that information and connect the results with the right samples that are in the system. And of course, the samples, as I showed you, are automatically connected upstream to those various source entities. And so ultimately, you have the ability to see, whatever information that you have captured for a particular sample, you can see how that's tied to a particular clone or sequence or whatever it is that you're ultimately analyzing. Let's see, I see a question in chat. Licensing. How does licensing work with regard to APIs and with regard to querying the LabKey instance directly? Yeah, so we do not charge extra for API access or anything like that. So we make, whether you are on-prem or cloud hosted, we have extensive documentation, examples, and support that will allow you to access the data directly through a variety of API languages. So, I'd say that the most common ones that people want to use are Python, R, Java, and sometimes JavaScript varieties. Querying any of your data through those just comes with any license of LabKey Biologics that you go with. And no, those querying accounts do not consume a license. So the implication in that question is that we offer per user pricing. That is true. And no, if you want to have a service account set up that manages those programmatic interfaces, that's perfectly fine. Thanks for that question. Okay. Any others? Those were great. Really appreciate your engagement. If not, like I said, there's a link that was shared in the chat. And we also try to make it pretty easy on our site to get in touch with us. I would love to spend a little time hearing more about your specific scenario and helping you see how that would work in the system displayed. Again, thanks so very much for taking a little bit of time this morning to join us and learn a little bit more. I'll look forward to future conversations. Thanks so much.